Hierbij open ik deze academische zitting, waarin Robert van Vorstenbosch het academisch proefschrift getiteld Translational Volatilomics for Digestive Health Advancing Early Stage Diagnosis and Disease Endotyping in het openbaar zal verdedigen. Mijn naam is Harry Schouten, ik ben emeritus hoogleraar hematologie en vandaag de prorector. Mag ik u verzoeken een samenvatting te geven van uw proefschrift?
Dear Prorector and members of the committee, and dear family, friends and former colleagues, thank you all for being here to share this special moment with me. Over the past few years, I've performed translational research into the use of volatilomics for digestive health, and in this presentation, I would like to tell you more about it. So, digestive health is a bit of a fuzzy term, for which I mean health of the colon, health of the liver, but also health of the gut-brain interaction. And before we continue, allow me to briefly elaborate a bit on those aspects. There are three diseases that fall into these categories, being colorectal cancer, irritable bowel syndrome, or IBS, and primary sclerosing cholangitis, or PSC. We are unfortunately all familiar with colorectal cancer, and as you may know, in the European Union, every year 500,000 people are diagnosed with this disease. That adds up to 4 times the entire city of Maastricht. And as we all know, detecting the disease at an early stage is favorable, and therefore countries have implemented national screening programs that consist of testing stool samples for the presence of blood, and if tested positive, a follow-up colonoscopy. Now, the problem here is that these initial screening tests are not very accurate, and therefore many people unnecessarily undergo this follow-up colonoscopy. IBS is a very different type of disease in which there's a disruption of the gut-brain interaction. Now, people who have IBS typically have a lot of abdominal pain, but there's also a change in stool consistency, so either diarrhea or constipation. And we know there are many factors involved in this disease, ranging from genetics to the gut microbiome to the use of antibiotics, but we don't really grasp the full picture. Therefore, it's really difficult to pinpoint a treatment to patients that have IBS. And what that means is that basically they undergo a sort of trial and error based approach in which they will leave out certain dietary components in the hope that that relieves symptoms, or they might go and try and talk to a psychologist with the aim of reducing stress to sort of restore this gut-brain balance. Now, the last disease is PSC, which is a rare liver disease, and it's very closely connected to inflammatory bowel disease, which is an inflammation of the colon. But exposure and the gut microbiome also play an important role here. Now, we can diagnose this disease using imaging modalities or liver biopsy, but the problem is that we typically do that too late. And therefore, people that have IBD indeed develop PSC. However, we detect that too late, and therefore the disease can progress to very dangerous stages, and those people in the end need a liver biopsy, a liver transplant, sorry. Up to 10% of all liver transplants are related to this disease, so it's a very serious condition. So these diseases all have their own challenges. So going back to colorectal cancer, it's a really good thing that we have this screening of the population, but of all positive screening tests, only 5 to 7% of individuals actually show to have the cancer. That means there are many unnecessary colonoscopies going on. Now, for IBS, we are really good at telling people they have IBS, but we're not so good at telling them what they can do about it. And for PSC, well, we can detect PSC, but we typically do it too late. Now, the result of these complexities is simply significant cost. So in the Netherlands, we spend on a yearly basis roughly 500 million euros on colon-related diseases, and for symptom-related diseases, it's almost triple the amount of that. And then again, it's because our screening tests lack accuracy. It's because we are not very good at assigning a good treatment strategy in case of symptom-related diseases. And for liver diseases, we detect the disease too late. Now, all of these diseases heavily depend on imaging modalities, but there's a world of opportunity that lies ahead of us that we may want to use to improve the situation, which is the world of omics. So some of you may have already heard of omics. You may be familiar with genomics, so the totality of all genes in your body. But there are other omics layers as well, including proteomics, so the totality of all proteins, metabolomics, the totality of all metabolites and chemicals, and exposomics, the totality of all the exposures that you undergo. Now, we focused on metabolomics and then more specifically the volatile part of that. So we enter volatilomics. Now, volatiles are small molecules that easily evaporate, so you can smell them, but you also can catch them non-invasively, making them very suitable for home-based monitoring or potentially direct readouts. Now, how does that work? Let's say you have a disease going on in your colon, chemicals are formed, and those enter the bloodstream, and you exhale them in your exhaled breath. Now, this chemical fingerprint that you exhale, we can link that to disease just in the same way as that we can relate your genetic fingerprint to disease. So then the question is, how do we capture this exhaled breath fingerprint? Well, on this slide, on the upper left corner, you see my former colleague Kim, who is sampling my exhaled breath. And what you see is that I'm breathing out into this mask. And what's basically happening is that my exhaled breath is being captured on these thermal desorption tubes. And these tubes have a sort of spongy material on the inside. That means that all the volatiles stick to this material, so we can trap the volatiles, meaning we can transfer them to the labs, maybe store them in a fridge, and then measure them later on. So let's say we do that. I bring my thermal desorption tube to the lab, and if we heat up this tube, all the volatiles can be released again. So they enter the gas chromatography column, where all the molecules are separated in time, and there we can identify them one by one using mass spectrometry. Now, as some of you may know, did not only look at exhaled breath, but also at fecal headspace. And for this, we used two sampling methodologies. First one being the microchamber, which is a method in which you sort of push all the volatiles through these same tubes again. So it's a bit like a chimney on a house. And the other method made use of Heisel probes. It's a bit more passive and a bit comparable to hanging a piece of tape from the ceiling and hoping that flies will stick to that. Now, both of these methods needed optimization, for which some of you donated samples. And we did not want to optimize towards already known compounds of interest, but rather we're interested in trying to optimize it in such a way that it would enable new findings later on in our clinical studies. So what we did is we maximized the number of volatiles we detected, but also reproducibility and biological variation. So what do we then see in your fecal volatile fingerprint? Well, we typically detect hundreds and hundreds of compounds. Those can include ethanol, for example. So I guess we will have a bit of ethanol later today, but your gut microbiome is also capable of producing ethanol, and that's actually really relevant in case of liver disease. Other compounds include dimethyl disulfide, which smells a bit like rotten cabbage, or butanoic acid, which is really good for your gut health, hexanol, which can signal inflammatory processes and oxidative stress, so it's bad for your gut health, let's say, and indole, which at low concentrations can smell like flowers, and at high concentrations smells a bit less pleasant. Then, having run all of our chemical analyses, it's now time for the data analysis. And one thing that is really important is that you choose a data analytical strategy that fits with the data characteristics at hand. And for us, that means we have very heterogeneous data, but also very noisy. And most importantly, that we want to make use of a sort of combined signal. Now, what does it mean? On the previous slide, I told you that butanoic acid is really good for your gut health. Hexanol shows stuff that's really bad for your gut health. And what we're interested in is a sort of health score that incorporates both of these compounds at the same time. So we want to have a sort of aggregate score with all of the good and all of the bad of all of the relevant compounds that we detect. Now here on the figure on the right, you see basically such a score represented by PC1, where if you move to the right, you enter a more healthy score represented by the blue dots, or the healthy controls in this case. And if you move to the left, you enter a more unhealthy score represented by the red dots, a pre-cancer stage. Now, over the years, we found that tree-based techniques fit with our data characteristics really well, and we use those for data exploration, data prediction, data fusion, and even something that's called one-class classification. For today, I want to skip all the technical details, and let's instead focus back on the clinical studies that we looked at. So to begin with colorectal cancer. Again, as I told you, so the problem was that this initial screening test is really not that accurate. So the question was, can exhaled breath volatilomics help? Now, short, we found that based on the exhaled breath fingerprint, we could correctly identify roughly 80% of the pre-cancer stages cases and roughly 70% of the healthy controls. And what we think is that the gut microbiome plays a really important role here. To put that a bit into context with what I told you previously, what we see is that, for example, butanoic acid, which is really good for your gut health, is present in decreased concentrations, and the bad stuff, showing oxidative stress and inflammation, is present in increased concentrations. Now, the second case study was about PSC, the rare liver disease, where we tried to aim for early detection. And again, the question was, can volatilomics help? Based on the exhaled breath fingerprint, we found that we could predict either absence or presence of PSC in 83% or 77% of cases. And based on the fecal volatile fingerprint, these numbers were 73% and 80% respectively. And if we combined both matrices, we had an accuracy of roughly 90%. What we also found was that the breath signal can catch the signal earlier than well-established clinical blood markers, showing that indeed it seems that we can go back to earlier stages. Now, there are a lot of, let's say, mechanisms and processes that seem to play a relevant role here, including exposure, but also the gut microbiome. And again, to put it a bit into context, we see ethanol again, we see the inflammatory markers and oxidative stress markers, we see indole that at low concentrations smelled like flowers, and limonene, which can smell like citrus fruits. The last case study was about IBS, and we asked ourselves initially quite a similar question: can we diagnose IBS based on a fecal volatile fingerprint? And the answer was yes. We found an accuracy of roughly 80%, but then we realized that if we simply ask patients, how do you feel, you will typically arrive at an accuracy of roughly 90%. So we sort of realized that we had been asking the wrong question, and instead of focusing on can we diagnose IBS, we think that the focus should be on trying to understand IBS, trying to understand what are the triggers of IBS patients and what can we do to help you live with this disease. Now, we explored that a bit in the final chapter 12. So to give a very brief summary, so we showed the potential to diagnose colorectal cancer and PSC. We started doing the same thing for IBS and then soon realized that we should really shift the focus towards better understanding IBS and trying to help people live with this disease in a better way. I would like to end off with quoting someone who recently inspired me, and this means something like stone by stone we can build a castle. And it reminds me of the perseverance and teamwork that we have in science, but also that we all have I think, a responsibility, but also the power to change the world. I think we all, whatever we do, we leave a footprint, so let's make sure that it's a positive one. And with that, I would like to thank you all. I would like to express my gratitude to the supervisory team, but also to Danielle Pach and Alex Mommers, our lab technicians. And I would like to acknowledge all the authors and first-year co-authors that I worked with. And with that, I would like to give the floor back to the pro-rector. Thanks.
Thank you for your clear presentation. The opposition will be opened by Professor Svetlov, Professor of Hepatic Inflammation and Metabolic Health at this university.
Dear candidate, I have to start by complimenting you. First of all, very clear presentation, but also mainly for your thesis, which I was very much impressed. And I'm not just talking about the quantity of the data that is outstanding, but also the quality that is also outstanding. I really like the idea that also you sort of like reflected, even in your introduction, on the new development. For example, on chapter 4, that you already said, like, okay, this is already not updated, and the full vision. And I really think you deserve compliments, and I would like to extend my compliments for your entire supervision team. So I would like to start by understanding more the potential of these VOCs, and in particular in relation to, because in chapter 9, you could actually differentiate between the IBD to PSC, which I thought it was quite impressive, and actually I was surprised by that. Because at the end, we are talking about cellular mechanisms, so to find really compounds that will be differential is, I was wondering, to what extent do you think that it will be a useful tool to differentiate between other, let's say, diseases that are digestive diseases, or is this like an exception that you can use it as a...
Dear highly esteemed opponent, thank you very much for the compliments and for an interesting question. I think it depends very much on the, let's say, clinical pipeline where you would like to use such a technique. So when I think about IBS, for example, then what I see is hopefully a future in which people can have a sensor, for example, at home, in which you breathe out, that will give you some clues about what kind of triggers you have and that you should try to omit. In case of liver disease, and specifically PSC, it's a bit of a different scenario, I would say. Here we really want to, with pretty high confidence, try to screen all the IBD patients. And I'm in that scenario, I don't think it needs to happen at home necessarily, but well, maybe it could. And what we are aiming for is...
Sorry, can I interrupt you for a second? Because now you are talking about how will you apply it, which is also very interesting, and I would like to go there. But I'm more interested into the principle of that from a mechanistic point of view. I mean, could you differentiate, for example, between different types of cancers, let's say colorectal cancers and HCC?
I don't know yet. So we don't have those studies yet. I do know that there are now quite big consortia going on where they are trying to answer these questions. And I think in the end, what is most important is how you're going to use it. So I agree with your question, but it also depends a bit on where you imply to, how you imply to use it. So in case of colorectal cancer screening, I'm not sure if it's such a bad thing, if in the end it's not necessarily colorectal cancer, but if we know that you have a different kind of cancer, then anyway that is enough reason to sort of look further for that. I think in general what we're after is not so much perfection, but more something that we can really apply in practice.
Or maybe in combination with other markers that could be more specific?
Yes, yeah, I totally agree. But to be more specific how well it can differentiate between different kinds of cancers, I don't think we have those numbers yet. So we would just simply have to see. Okay.
And metabolic diseases are usually they have a very dynamic nature, right? So how like the diet or many things that will influence it quite fast. Actually, it can happen really quite fast. How do you make sure that you monitor it in a good way, or that you get rid of all this noise? I mean, does a patient has to, I don't know, test it several times in a row? How does it work?
Yeah, really interesting question. Thank you very much. And this is a question that has been popping up over the years quite a few times. How we have regarded it is that differences in diet are a sort of background variation that you'll have anyway. Because the one thing that we cannot standardize is that everyone will have exactly the same diet. What the field is moving towards is that we use so-called EVOC probes. So a substrate that you basically take and we measure how well you metabolize that. And by doing that, you can sort of artificially really increase the levels of the relevant VOCs you're interested in, and thus also sort of go beyond this background biological variation to which diet also contributes.
But also then monitor the time, that passed from. Okay, yeah, that is really interesting. And you already hinted on the home use that you started to discuss it. So how does it work in practice? What is the economic, I don't know, how much do you have to invest in actually giving it to patients? How will it work? Will you borrow it for some time, or is it regular that patients will have? How do you see that in the future?
Yeah, also again, a very interesting question, and obviously we are not there yet. So it's difficult to answer that in a lot of detail. But to be fair, if a patient has to come back to the hospital several times and has to be seen by a specialist, and maybe you have colonoscopies or whatever, then that is also really expensive. I think it depends very much on the use case. So I think let's say the first sort of stop to go to is where you try to control everything really well. So maybe start doing these breath tests in the clinic. Maybe that in the beginning will not be super cheap yet, but at least it will be cheaper than a lot of alternatives in the clinic that are right now there.
And for early diagnosis, I can see why it's so efficient. Do you think that it will be, let's say, accurate enough to also monitor progression of diseases? So I'm not just talking about flares, that's clear, but really just progression?
Depending on the use case. So in case of, I think in case of liver disease, probably yes. But I don't think this will be necessarily the case for all diseases. So we just have to see what makes most sense. I think in the end, the aim is not to just use breath analysis everywhere, but to make sure that the patient is helped in the best way. And maybe breath analysis for some diseases will just be used as sort of screening tests, after which they might receive other kind of medical tests and imaging. And maybe for some cases for liver disease, just to sort of see the progression over time, it can be a really interesting tool.
And how will you set up such an experiment to see a progression over time, if you want to test it?
So again, going back to these substrates, so, well, let's say the dream would be that patients at home would have a small sensor in which they could breathe out. They take a sort of pill which contains this substrate, and over time you measure your exhaled breath fingerprint, let's say 6 times, so that you know how fast you metabolize that substrate. And this you could repeat over time.
So with the
once every 3 months or 6 months, or depending on the situation.
So with the EVOCs, like that's the direction. Yeah, okay. So I have another question. I would like to ask one of the PANIF to read the Stelling 10.
In light of scientific challenges our society faces, the solitary structure of a PhD trajectory should be reconsidered. Shared and collaborative PhD projects are likely to be more efficient, successful, and mentally gratifying, according to this thesis.
So I have to say, I was very much amazed to read this Stelling, because it's the first time I heard about something like this, and I'm asking you because I agree with you, so to start up with this, but I want to hear more about it. What is your vision about that, and why? And
thank you again for your interesting question. I guess I was very lucky to have worked with several people who have, coming from the clinics, for example, who have way more knowledge than me on liver diseases or gut health or all of that. And I think the challenges that we face are too big for one person to know everything. I also don't know everything. And the very nice thing was, I think that if you collaborate, then I can focus on the data structure and what are we looking at and trying to understand the numbers. And then other people can, let's say, do their part. And I really think working together, we can, well, scientifically achieve more, but it's also simply more fun and mentally, I think, better, not having the feeling that you have to do it alone, but that you do it as a team. So I'm not sure if that answers your question, but
no, it does. I would love to talk more about it during the break. Thank you very much. I'm happy with your answers, and I bring back the word to the co-rector.
Thank you. The opposition will be continued by Professor Heren, Professor of Molecular Imaging at this university.
Dank u, meneer de pro-rector. Dear candidate, I would also like to extend my compliments to you for this very heavy book with lots of data and lots of different approaches. And of course, I want to compliment your supervisory team as well with this achievement. We heard you talk a lot about the different profiles that you associate with the different diseases. Now, I looked through your thesis and I found the word correlation about 50 times. I looked for causes, for causation, and unfortunately I only found that 11 times, and then not really in the context of a cause of the profiles. So this dilemma of correlation versus causation, I want to question you about. What do you think is more important from a patient diagnostic perspective? And can you elaborate on your choice a little bit?
So I think from a patient diagnostic perspective, the most important thing is that we have something that works. to begin with.
Okay, but now you're avoiding the question. I'm talking, what do you think, correlation or causation?
Yeah, I think it's a really interesting question. So from an epidemiological point of view, obviously we're interested in causation as well. And I do think it would be good if the signal that we catch, that we're sort of sure that it really links to causation. On the other hand, correlation is not useless when we think about diagnostics and prediction. So let's go back to the colorectal cancer case. Maybe the gut microbiome is not changing, maybe that's not causing the cancer, but maybe it changes because there's a cancer present. For us trying to catch that signal, it doesn't really matter much why that shift is caused. As long as we can sort of catch that signal that belongs to this sort of cancer status.
I would argue, I mean, on the one hand, I agree. On the other hand, as a fundamental scientist, and if I think about different treatment options, and particularly the heterogeneity of the gut microbiome, that might influence many of these signals that you interpret. Mm-hmm. So if I would know the cause of these changing profiles, would that not be much better from a diagnostic perspective? Because then not only can you tell whether or not the patient is suffering from PSC or IBD, but actually treat them better.
Yeah, I agree with your question, but then you're also asking different questions. So then it's about what is the best treatment option, maybe medication. I fully agree that that's also a very interesting question, but it's a slightly different one.
Okay, now if you go that route, then I'll go to my next page. I have a different question for you, because we're talking about correlation, and in one of your chapters, you actually discuss quite a lot of machine learning and deep learning approaches. And you there talk about the fact that these machine learning approaches are prone to overfitting. And you mentioned that a number of times, but you don't really define it. So before I go any further, can you please define for me, what do you determine as overfitting? Or how do you define that? Sorry, not how you determine it, how do you define overfitting?
Thank you again for your interesting question. In the end, what we would like to have is a test that if you go to a new population, that it will still work. So not that it's something that saw a pattern that's only there in that current data set, but really something that generalizes to new unseen data.
So and your data, how in what category does that fit then?
So you mean in terms
your
of
analysis, yeah.
So what we so what I would say is our data sets are relatively small, so sort of get what you are getting at. At the same time, so we are really conscious about this problem. So what we try to do is we try to determine independent test sets that were not seen during the training phase, and we try to work with that. So within, let's say, the limits that we have, we do try to deal with that problem. But I fully agree with you that we do need more studies to test these methods on new unseen data. I don't think we have the illusion that if we take these models now and we go to new unseen data, that they will magically still work. Mm-hmm. I think you have to look a bit more critical.
Will they become better in your mind? Because that's actually what you're discussing, is exactly where I wanted this discussion to go, is the challenge of larger patient cohorts, because you have a relatively small study. Within your small study, you see these correlations. But if you now increase the number of patients in your cohort, let's say do a multi-center study where, you know, with different diets, different genetic makeups, different exposomes, how well would that still hold up? Because in those cases, you would increase the heterogeneity of your data set, and, or in your terms, the dirty data.
Yeah, so what you need is to go stepwise. So most of the studies that we performed are very exploratory in nature. So you should not go and just transfer these models now to new unseen groups. So what you want to do is check, okay, what kind of patterns did we see? Can we try to measure that in new studies, but can we also check what other people found in their own smaller studies? Can we try to implement all of that information in bigger cohorts and then see, you know, what kind of patterns do we see? So, you know, I always compare it with going to the moon with a rocket. Like, we can go there. It doesn't mean that we found everything in one time. We need to go up and down and learn from that process, and then...
Right. So science is an iterative process.
Yes.
Okay, let's change topics. Very interestingly, in the introduction, you actually talk about LCMS. And you're out of luck because I'm an omics guy, I like LCMS, and there you state LCMS is less sensitive. And I understand you're into gas, so you use gas chromatography. But for smaller molecules, one very important aspect is the identification purposes. And you talk about that a number of times. In fact, you state for smaller molecules, the retention indices are trivial. You talk about LCMS and provide sufficient information for identification purposes. That's one of the sentences on page 59, if you want to check. Can you explain to me, if I give you a random retention time, 8.9 minutes, let's say something, do you then know what molecule that is?
Are you talking about GC or LC?
No, LC. Yeah, you talked down LCMS. So I want to convince you that there's more to LCMS than there is that you give it credit for.
It all depends. So I think we have to be open and honest that both LC and GC have their problems with compound identification, especially when we move back to lower concentrations where we see a lot of, let's say, variation in mass specs. What we know is that GC is then relatively more comparable across platforms, let's say. So on the one hand, I want to answer yes to your question, like yes, we can identify everything easily. On the other hand, I think this is going to be one of the most crucial sort of things that we still need to do in science to make sure that these, well, in this case, mass specs, become more stable so that we can improve.
Okay, now I'm going to challenge you, because have you ever heard of QA/QC protocols?
Yeah.
Have you ever looked at how clinical diagnostic labs use their QA/QC protocols in their mass specs and how they very accurately, quantitatively, and reproducibly determine all kinds of blood profiles?
No, I'm not familiar with how that goes in clinical labs, but I'm sure that the scenario is a bit different than what we are looking at, where what we are doing is really exploratory purposes, untargeted metabolomics, trying to catch any signal that might be there. Whereas clinical labs often have a much more targeted approach to stuff, and if you know what you're looking for, then your life becomes quite a lot easier.
Okay, I'm looking at the project. Can I ask one short question? Because you perfectly touched upon the targeted versus the untargeted approach. And I want to challenge you on that. On page 168, you talk about where you develop your extraction methods and you test them. You talk about 26 selected VOCs and you use a design of experiment approach to optimize your capture methods. Then in table 3, you conclude that, okay, we see 130 VOCs with one method, 150 VOCs with another method, but then underneath in the figure, I only see 27 peaks in your GC. What happened to the 120 or 103 other VOCs that you apparently discovered, although putatively identified, because I don't think you've done tandem MS?
So, thank you again for this question. I think in this case, it also just simply has to do with communication. So you want to communicate sort of clearly about the findings that you have. And I'm not sure if I would assign 128 peaks in this chromatogram.
You did in the table.
That would clarify the picture a lot. And what we also know is that we are not expecting to find all VOCs in all samples. So there's a lot of variation.
You're slowly avoiding the question because you don't tell me where the 103 other VOCs are. But maybe that's something for which we'll save for the ethanol. Thank you, and I'll give back the word to the prorector.
This was not a short question. And the risk we are running was a very long answer. Yeah. Okay, I should have learned from this. The opposition will be continued by Professor Di Francesco, Professor of Breadth Analysis at the University of Pisa. Thank you for coming to Maastricht.
Thank you for inviting me. And it was a pleasure reading your thesis, one of the best theses, PhD thesis I have read. I have the pleasure to be revising. My compliments. I've seen that you have, in the breath community, there is a lot of discussion about how to improve and how to bring this to the hospital and why despite of many decades of work, we haven't been successful in achieving real tests. So one of the main discussion is about the standardization of the measurements, and sampling is an important part of it. I've seen that you have used the receiver from Allstone to sample your breath and in your patients, and so I would like you to explain why such choice and what are the advantages and the limitation that you can see in this approach compared to what is described in the literature.
Dear highly esteemed opponent, thank you for your compliments, and also thank you for coming all the way here. We very much appreciate it, I think also on behalf of the supervisory team. If I understand your question correctly, you would like me to elaborate a bit on the advantages of the receiver device. Yeah. So before the receiver, we were using Tetler bags. And to be really honest, we would often also sort of reuse them. So we did not use them only once, but multiple times. And in Tetler bags, you just capture the entire exhaled breath fraction. So it's sort of the dead volume, but also the place where the interchange between blood and breath happens. And because of that, there's a strong dilution going on. Now, what we find with the receiver device is that there are quite some advantages. So they aim to capture only that exhaled breath fraction where this interchange is happening. With that, let's say all of the exposures that are not part of your biological system are sort of reduced. But we can also sample for longer, so we can capture larger amounts of these VOCs we're interested in. What it also does is it supplies clean air, so the influence of room air contamination is also smaller. So those are a few examples of the advantages of the receiver device. Does that answer your question?
Yeah, and it's ready to use.
Sorry?
It's ready to use. Yeah, you don't need to develop technology because it's a commercial device.
It is something that can be used relatively easily in the clinic, so that's good. On the other hand, it does not really allow for home-based sampling. So, for example, a while back I was thinking about an epidemiological study where perhaps it would be nice to sample exhaled breath. Well, we cannot give everyone a receiver device, so in that sense it's not such a good fit. And sometimes it can also simply take quite a while to sample a patient. So it can still take 15 minutes. We've seen 20 minutes, and normally after that we sort of stop because then a patient also becomes too much. So I do think there are still a lot of developments that also can happen in terms of this.
Okay. In your thesis, you introduced unsupervised random forest, SIMCA. And why was one class model needed, and how this your hybrid method outperformed the other existing approaches? I think this is one of your major contributions. I think you're quite proud of it.
Yeah, thank you very much. I think this also relates back to questions of a previous opponent, and this has to do with trying to get results that are as close to the truth as possible. So we can be a bit unfair and always build binary classification models, but sometimes we have classes that are just undersampled, maybe we don't have enough cases, or maybe those classes are very heterogeneous. And what you sometimes see in the literature is that maybe there's a class of 50 cases and the other class is 6, and people will build a supervised machine learning method, or they will use that to make predictions. And we think that is talking about overfitting, but that's really not fair, because how can you validate that? You cannot even add enough variation to the training phase to really make it work. So then there's a lot that you can do. So you can build artificial samples based on the samples that you did have. So let's say we are now creating a lot of artificial samples based on those 6 samples that we have in our set. But if you do that, the information of those original samples is still present in those artificial samples. So you're basically just fooling yourself and you're adding the information twice, basically. So we think that that is inherently a wrong approach, and what you can do, and that is what has been going on in the field of chemometrics for a while, is to build a model based on only one class. And we tried that, and it worked really, really badly on our type of data. And that made us wonder, okay, what can we do to maybe improve that philosophy? We agree with the philosophy, but the methods are not working. So that is sort of how we came about the unsupervised random forest SIMCA.
Okay. And other question is you have several chapters in your thesis in which you describe methods for data analysis, very powerful methods for data analysis, and you include machine learning, deep learning, and so all these methods rely on a large number of parameters that you can play with to develop your model. On the other side, you have, and this is not just a limitation of what you did, it is more general. If you go through the literature in breath analysis or also in the headspace of fecal samples, you have limited number of patients, very large variability, intra-individual variability, and inter-individual variability. And at the same time, we suspect that some of these pathologies have a dynamic behavior over the time, and we rely typically on cross-sectional studies because many times we don't have the resources to perform longitudinal studies that would be needed to learn more about patients. So, and you mentioned that this risk of overfitting, but the fact is that we need to consider also that the medical class is quite conservative, and we are not really successful in bringing this technology to the clinic. So the question at the end is, I would like you to comment on my previous statements, and I would like to know your view on this, and what is really needed to make something that would be a method that would be reliable for the patients.
Thank you for this very interesting question. To start at the beginning, so we use these machine learning methods not because they're powerful, they are powerful, but that was not the reason. The reason is that they make use of mechanisms that fit with our type of data very well. And I think that is the sort of the crucial aspect. And that is also where the answer to your question lies. So our type of data has a lot of missingness. There are all kinds of artifacts in our data. And I think one of the reasons that this is not ending up in the clinic yet is because all of those artifacts and reasons, and I think the field is doing a very good job. There are a lot of improvements, but in order to get it to the clinic, it has to be something really robust. Preferably, you would not want to need those very powerful machine learning methods. Maybe you want to use them because they're powerful and will give you good predictions, but you don't want to use them to sort of correct for artifacts in the data, for the missingness and the zeros. I think what we should do and aim for is to improve the quality of the data, that you don't necessarily need those machine learning models to sort of improve that problem, and to really create very robust data that has been shown in larger cohorts, and then go back to the clinics and say, hey, we can really very reliably use breath analysis. And so far, it's not there yet. It's not that reliable yet. And if we are really honest, we've had so many problems in the lab with the GC and with whatever. You cannot sell that to the clinics yet. So the most important thing we need to do is to create something that's really robust, that can run 24/7.
Okay, I'm satisfied from your response. I give back the floor to the director.
Thank you. The opposition will be continued by Dr. Folcarelli, Senior Data Scientist at Corbion Purec in Gorinchem. Thank you for coming to Maastricht.
Dear candidate, I would like to congratulate you on this really nice work. It was a pleasure to read, and I think, you have a really deep knowledge for both experimental part and, of course, data analysis and chemometrics. So my first question is on chapter 7 as well, on the new method you developed, the unsupervised random forest SIMCA. I understand your choice to go for one classifier, so class modeling, but I was wondering if you try to do some, perform some more discriminant analysis, also to benchmark with your approach. So can you elaborate a bit on that, if you apply some discriminant analysis as well?
Dear esteemed opponent, thank you very much for your question. I'm thinking about what is the best way to answer, because yes, we did compare it, because most of the data sets that we used in that study we were quite familiar with, because those were the sets that we did build, often binary classification models. In this chapter, we did not want to compare it that directly, because it also sort of raises a very confusing image, because one-class classification models are not competing with normal binary classification models. If you would have data that also fits with a normal binary classification, then you would go for that, because it's more powerful. You can do you can extract feature importance and do feature selection, which is way more problematic in the case of one-class classification. So in that sense, we felt it's not really a very fair comparison, so we aim to just compare it to other already existing one-class classifiers. We did, in the end, compare the, let's say, the importance of the features extracted by our method with random forest, for example. We did not find a significant difference, but well, I have to be honest that the uncertainty there was also relatively high. And, you know, in the end, it is still something that one-class models are only based on one class. So I always wonder how well it's possible to do really any kind of feature selection in that. I think, depending on the scenario, sometimes may be possible, sometimes not. But anyway, sorry, concluding, it's not such a fair comparison.
Yeah, okay, thank you. But maybe a combination of the two approaches could make the whole analysis more powerful. So maybe it's something that you could do.
Yeah, sure. So I think, well, you know Beata Wolczak. She tried to do something similar where I'm not quite sure now if she used PLS or random forest, but you could run first random forest and then do the SIMCA approach on those proximities as well. So that's also an option.
Okay, thank you. And then about, random forest proximities. So this may vary based on the hyperparameters that you have in the method. So did you do any optimization for that, or what you will advise to do?
No, this is possible. We didn't do it, so we used it a bit naively. I think what would be good is to have a sort of protocol on how to do this, because I find it always a bit difficult on how to optimize within a one-class classifier. If we're gonna use unsupervised random forest for feature selection based on one class, are we gonna throw out features that might actually be relevant for discriminatory purposes, or what are we actually filtering out now? And I always find that becomes a bit fuzzy. So I think this is also an area of research where, you know, extra attention should go towards too. There are a lot of optimization strategies, so the compliant approach or the rigorous approach, so those can be used here as well, but I think it's very much dependent on the scenario, on the very specific scenario you're focusing on. So it's a bit difficult to give a very straight answer to that question.
And another one, the model that you applied are mostly on a population level. So I'm wondering if you can a bit comment how you will change the approach and pipeline also in terms of chemometrics model to apply towards more personalized medicine, so more on the individual level, and because that can be useful indeed for a more personalized medicine. So yeah, how you will change the approach, and if you know any chemometrics model that will be more suitable in this case.
So is your question about how I would update the current technique in case of personal or different methods?
Well, it's open. Either if you may want to adapt or maybe use other methods.
I think there may be multiple approaches. One of the challenges will be that we think that these methods need a relatively large sample size. So that will be the best thing. As soon as you go fully personalized, which I can totally imagine that you maybe have repeated samples of an individual over time and you only use the individual data to build the one-class classifiers. In case of IBS and liver disease, maybe a very interesting approach. In such a case, I find it difficult to predict how many samples you would need. You could use the same features that we find for the population, then sort of update the model towards the individual. I guess what would happen is that you first start with the more population-based models, and while a patient gives more samples, then maybe you can sort of update it to a more personalized model in this case. But as with anything, maybe it's best thing to first start to see if we can make use of these population-based effects and see what do we find there, and then if that's interesting, maybe we can sort of further update it. And if that's not interesting, then maybe that's a sign that's not worthwhile.
Okay, thank you.
Thank you. The opposition will be continued by Dr. Koek, gastroenterologist, hepatologist at Maastricht UMC+.
Thank you, Pro-Rector. As a challenge, I want to change to the Dutch language. Maybe some people still understand Dutch, so therefore I was not prepared on English. And you see my English is bad, so I changed to Dutch. Geachte promovendus, een zeer dikke proficiat voor u en uw promotieteam voor uw zeer uitgebreid wetenschappelijk onderzoek beschreven in uw volumineuze proefschrift. Vele jaren geleden hebben we in Leuven bij IBD-patiënten X-Health NO als predictor voor informatie onderzocht. De test was specifieker en sensitiever voor longziekte dan bij de IBD-patiënten en wordt ook niet meer gebruikt. Een visionair hoogleraar aldaar stelde dat onderzoek in de ochtendflatus veel informatie zou geven over de gezondheid van de patiënt. Hij had echter geen goede methode ontwikkeld om dit te kunnen uitvoeren. Nu, anno 2025, presenteert u hier een uitgebreid goed onderbouwd onderzoek naar VOC's en is volatilomics een serieus onderzoeksgebied geworden. Ik wil u over hoofdstuk 4 en 5 wat van gedachten wisselen. Ten eerste stelt u, heel mooi in dat onderzoekje, die 382 IFOBT positief en coloscopisch gecontroleerde patiënten, dat uw VOC veel preciezer kan aantonen dat een patiënt een coloscopie wel of niet moet krijgen. Dat vind ik natuurlijk als MDL-arts jammer, want dat is mijn core business. U heeft waarschijnlijk een coloscopie al meegemaakt om dat eens een keertje te beleven. Maar mijn vraag is eigenlijk, hoeveel van die IFOBT's worden er jaarlijks gedaan in Nederland?
Best hooggeleerde opponent. Dank u wel voor uw complimenten. En ik moet u toegeven dat ik dat eigenlijk op dit moment even niet scherp voor de geest heb.
Oké, dat zijn er 1,5 miljoen bijna. Hoeveel kost het? Heeft u daarover nagedacht?
Als vervolgens de vals positieven zo hoog zijn dat al die mensen door worden verwezen naar colonoscopie, dan is dat best wel duur.
Maar ik bedoel de test zelf.
De test zelf zou niet zo duur zijn.
Nee, dat is correct. Die varieert van een tientje tot €38. Hoeveel kost het bepalen van een VOC?
Dat heeft de potentie. Ik denk dat dat uiteindelijk ook zo op de 50 gaat uitkomen. Dus dat is een beetje duurder dan de test die u omschrijft. Maar dat zal zeker, denk ik, ja.
Oké, maar 1,5 miljoen. Dan kunnen we dat vergelijken en dan kijken we wat de balans is natuurlijk. Want dat is natuurlijk wel essentieel. Want u wilt het verkopen aan de policymakers en aan onze gastroenterologische vereniging dat wij overgaan naar ademtesten.
Ja.
En hoeveel laboratoria zijn er in Nederland die ademtesten goed kunnen uitvoeren?
Nou, die zullen er denk ik wel komen, want ik ben natuurlijk straks ook klaar en die ga ik dan allemaal oprichten.
Oké. Ik zie al een business case komen.
Ik denk dat u de situatie een beetje versimpelt, want wat nou uiteindelijk natuurlijk zo duur is, zijn al die specialisten die colonoscopieën moeten uitvoeren. Die raken gewoon helemaal overweldigd door het aantal mensen dat die colonoscopieën nodig heeft.
Nee, maar we laten het de verpleegkundige specialisten doen, die zijn weer goedkoper.
Ja, maar alsnog belandt u wel in een logistiek zeer moeilijke situatie, omdat er gewoon zoveel mensen nodig zijn. Omdat zoveel mensen die colonoscopie nodig hebben en de capaciteit daarvoor gewoon echt moeilijk te vullen is.
Ik hoor nu een manager die mij dadelijk gaat regelen hoe dat georganiseerd gaat worden. Dat hoor ik eruit. Maar toch ben ik nog niet overtuigd van het feit dat die VOC's het zullen worden. Ik ben een voorstander van VOC's, dat sowieso. Want als je naar een colonoscopie... Heb je een colonoscopie gezien een keertje?
Een simulatie.
Oké, is goed. Dus geen colonoscopie. Maar hoe groot is een polyp? Want als je nagaat dat een polyp dit is... Ik heb er vorige week nog een paar uitgehaald, die zijn superklein. En dan kan je in de uitgeademde lucht detecteren dat daar een polyp zit. Of een carcinoom in situ of een beginnende maligniteit. Als je kijkt, het kolon is 80 centimeter lang en er zit zo'n mini dingetje.
Ja.
En u haalt het er zo uit, dat vind ik toch knap. Of is dat misschien toch niet waar dat u dat kunt detecteren?
Ik denk dat het heel goed is om daar heel kritisch op te blijven. En dat relateert een beetje weer aan de eerdere vragen. Dus wat wij eigenlijk zelf denken is dat als we nou focussen op het microbioom, dat zijn nog veel meer cellen dan dat we zelf cellen hebben. Als dat microbioom verandert omdat iemand een polyp heeft, of andersom, dan kunnen we misschien proberen om dat signaal op te vangen wat bij dat microbioom hoort. Tegelijkertijd zien we nu ook resultaten van Alstom Medical uit Engeland. Bent u geloof ik bekend mee? En die EVOC-probes die zij hebben ontwikkeld en die nu voor die levertesten worden gebruikt, die hebben zij heel succesvol voor longkanker ingezet. En ze zijn nu verder aan het kijken of ze ook voor andere kankersoorten datzelfde kunnen doen eigenlijk. Dus persoonlijk ben ik het met u eens. Ik verwacht niet dat we zo'n hele kleine polyp alleen maar dat signaal kunnen vangen. Ik denk dat we breder moeten kijken om ook het signaal te kunnen vangen wat bij het microbioom hoort. Maar er zijn wel degelijk signalen uit het veld dat we vroegtijdige kanker wel kunnen oppakken. Maar in dit geval ging het over longkanker. Maar...
Ik zeg altijd, ik vond het iets wat ik in uw thesis heb gelezen, l'orchestre du microbiologique. Geen Brahms, geen Shostakovich, geen Bernstein, maar is in feite een 12-tonen metabool aspect. En dat is moeilijk te dirigeren, want die microbioom is toch vrij moeilijk te dirigeren. En ik denk dat die metabolieten, die gaan allemaal in het portale systeem en komen in de grote orgaan, de lever terecht. Dus meet ik niet het metabolisme of het dysmetabolisme, niet vanuit het microbioom, vanuit de darm zal ik maar zeggen, maar uit de processing die in de lever plaatsvindt. Wat is daar op u... Wat denkt u daar zelf van?
Daar ben ik helemaal met u eens. En dat is precies het signaal dat we willen vangen. Het moeilijke aan metabolomics op basis van thesis is dat patronen die je daar ziet, voor example in short-chain fatty acids, dat je dat niet per se ook in je eigen systeem terugziet. Dus wat heel belangrijk is hoe goed je die stoffen ook zelf opneemt. Dus er zit een belangrijk verschil tussen. En waar het op lijkt is dat een van de mooie dingen aan uitademingsluchtanalyses is dat iets stabieler is. Dat het iets meer zegt over wat zit nou eigenlijk in jouw lichaam en niet alleen maar over wat gebeurt er in jouw darmen. Omdat daar zoveel variatie ook zit. Ik denk dat dat juist op een bepaalde manier een soort stabiliteit kan geven aan de metingen.
Maar meet ik dan niet het geprocesseerde gebeuren wat in mijn lever plaatsvindt, komt in de gewone bloed terecht, komt in de longen terecht en dan adem ik uit.
Ja.
En niet zozeer, die darmen, dat was wat die hoogleraar zei. Die flatus, dat is een echte weergave van wat er in die darmen plaatsvindt. Maar niet wat er geprocessed is in de lever en dan uitgeademd wordt.
Ja, u moet heel snel zijn, want ik weet gewoon niet wat u wil weten.
Nee.
Nou, die discussie gaan we niet aan, wat u wil weten. Robert van Vorstenbosch, de gelegenheid voor het verdedigen van die proefschriften is voorbij. De promotiecommissie zal zich nu voor beraad terugtrekken. Ik verzoek u en uw gezelschap onze terugkomst en de uitslag van dit beraad in deze zaal af te wachten. Robert van Vorstenbosch. De commissie hier aanwezig heeft zich beraden voor de kwaliteit van uw proefschrift en de wijze waarop u het proefschrift heeft verdedigd. Op grond van haar positief oordeel en gelet op de examens die u vroeger heeft afgelegd, heeft zij besloten u het gevraagde doctoraat toe te kennen. De hoogleraar professor van Schoten is gemakkelijk u op de gebruikelijke wijze met deze waardigheid te bekleden. Het woord is aan uw promotor.
Belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan, eerlijk, zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Dat beloof ik.
Mooi zo. Krachtens de bevoegdheid, ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hier bij u, Robert William Richard van Vorstenbosch, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens wet en gewoonte zijn verbonden. Ten bewijze hiervan overhandig ik u nu de bul door rector, secretaris en overige leden van de promotiecommissie ondertekend en met het grootzegel van de universiteit bevestigd. Dokter van Vorstenbosch, van harte gefeliciteerd. Beste Robert, van harte, proficiat. Geweldig wat je gedaan hebt. Vandaag een fantastische prestatie over de afgelopen jaren. Een fantastisch proefschrift geschreven. Je mag hier heel trots op zijn. In ieder geval, ik ben heel trots op je. Zoals gebruikelijk zal nu de laudatio gegeven worden. En dat zal je begeleider Akkie doen. Akkie, het woord is aan jou.
Dear Dr. van Vorstenbosch, please accept my warmest congratulations. And I think now I can say, dear Robert, it is my honor to be the second one. To say, I'm so happy to know that you have reached your goal. You truly have proved your self-dedication and determination. It is with great honor and pleasure that I stand before you to deliver this laudatio, celebrating the commitment, insight, and the impact you have brought to the field. I want to personally congratulate you for being such a huge success and setting a good example to other colleagues. Together with Professor van Schoten, your promoter, Dr. Mujagic, your co-supervisor, we are proud that you have gained your PhD degree after all the hard work and challenging you face along the way towards your PhD degree. Remember your study, data, and the thesis. Every time you are thinking that you were almost there, but you often did not see the end. Well, look at you now. All now has paid off. And again, congratulations. Dear colleagues, friends, and guests, today we celebrate very special PhD candidate, Robert, someone who has been, in many ways, quite one of a kind. Many Mondays mornings began the same way. Robert walking in the office, dropping into the chair and asking his colleagues, and who was your weekend? And then, of course, there would have been a gezellig chat about the weekend. Honestly, how many young researchers his age starts their week by checking in like that? It shows something essential about him, his warmth, his consideration, and his ability to make working together feel human. Let's not forget our ever-charming, that aging masterpiece, almost retired GC MS, that spent more time in maintenance mode than in analysis mode. Robert would be the person who would be always helping. We could ask him for help. Daniela, Alex would be asking him, and he would be always there to help. Even if he couldn't fix it, together with Alex, he would be sitting next to Daniela and support her just to share her frustration about our lovely GC mess. The teamwork spirit was one of your trademarks, Robert. You love to say, we are a good team, and you meant it. Of course, emotions were also part of the journey, a lot of emotions. There were moments where things felt overwhelming, when choices, words, or action needed to be thought through carefully. But what made Robert stand out was his openness. He never shut down feedback, never avoided difficult conversations. Instead, we talked about how he saw things, how we saw things, and step by step he grew. And in the end, it did work out. The thesis you produced, it's amazing. I still remember our first journey to Poland. That was still before the COVID time. At the start of your PhD, back when you were taking your very first baby steps in research. And now, a fully grown scientist, standing tall, confident, and more than ready for what comes next. Robert, you were a PhD who did not wait for others to solve things. You went to the lab, you analyzed your own data, you confronted every challenge you had. In between, you were also helping others with their data, experiment, or taking over teaching duties when it was needed. 400 fecal samples to measure manually. Can you imagine? Well, Robert did. Most people would panic, but not Robert. Robert's response was simply, consider this done. Nothing was impossible to you. And he was never afraid to go beyond his expertise. Truly admirable. And yes, his desk was a glorious mess. There was a moment when he brought a flower just to make it look better for his office mate, because that is who he is. He makes ordinary things special. Even choosing tea with him became an event. He would look at the tea selection and ask, so, what do we drink today? And as if we were selecting vintage wine. Robert, you were always incredibly enthusiastic, full of passion for your project and equally capable of overthinking every detail. And then often there were those moments when you sent WhatsApp messages so long they made my brain politely ask for a coffee break. But important thing is that once you talk, the doubts cleared and the determination returned. You care deeply, not only about your work, but about people around you, about doing things the right way. You stood up for yourself where something was unjust, like the time your work was mistakenly omitted at the conference. You defended your contribution with confidence and dignity. Most of all, you made people smile, many times. You created warmth and connection in small everyday moments. Robert, today we celebrate not just your academic achievement, but the person you were through this journey. Dedicated, emotional, humorous, passionate, caring, ambition, and uniquely you. Therefore, I would like to congratulate you again. Also to your parents, congratulations, you should be proud. And your partner, Jasper, congratulations, they can be proud of you. We are proud of you. Congratulations, Robert. You have truly earned this moment. Thank you.
Dr. van Vorstenbosch, ook ik wil u graag feliciteren en ik mag het ook doen namens de hele universitaire gemeenschap van Maastricht University. Geweldig werk, u bent deel van een hele belangrijke ontwikkeling in de zorg. Elke scopie die we kunnen voorkomen is denk ik mooi meegenomen. En uw testen mogen best wel wat geld kosten, want een colonoscopie kost toch iets van €1200. Sommigen zeggen dat ze het goedkoper kunnen doen, maar ik heb de rekeningen gezien. Dus ik wens u heel veel succes toe in uw verdere carrière en het allerbeste. Ik nodig u dadelijk uit om zich te begeven richting de refter, om de jonge dokter te feliciteren met het behaalde doctoraat. En daarmee sluit ik deze academische zitting.